Bismillahirrahmanirrahim. Assalamu alaikum. Dear students, I am Dr. Mahmuda Sultana Shumi, Associate Professor, Department of Anatomy, Sakina Women's Medical College, Jashore. Today I am going to discuss about the histology of lungs. Structure of lungs, there are three codes. These are serous coat, subserous coat and intrapulmonary substance. Serous coat, that means pulmonary pleura, and subserous coat consists mainly of elastic fibers, and intrapulmonary substance consists of a system of branching air tube, that means interpulmonary bronchial tree. These are bronchi, bronchioles including terminal bronchioles, respiratory bronchioles and alveolar ducts, which open into atria and in turn atria open into alveolar sac and alveoli. A microscopic section of the lung shows a network of empty space, that means alveoli, surrounded by thin wall intra-alveolar septum and contains different orders of intrapulmonary bronchial tree together with blood vessels, lymphatics and nerves. So, the lung alveoli giving a honeycomb appearance in section and lined with simple squamous epithelium. The adjacent alveoli shares a common inter-alveolar septum. Bronchus contain pieces of hyaline cartilage. Each respiratory bronchioles gives rise to an alveolar duct and numerous alveoli. Bronchi and bronchioles, that means terminal and respiratory bronchioles, are scattered in the structure. Alveoli are sac-like evagination, about 200 micrometer in diameter of the respiratory bronchioles, alveolar duct and alveolar sac. Alveoli are responsible for the spongy structure of the lungs. Structurally, alveoli resemble a small pocket that are open on one side, similar to the honeycomb of the beehive. Within this cup-like structure, oxygen and carbon dioxide are exchanged between the air and the blood. They are separated from each other by interalveolar septa, which may contain one or more alveolar pores. An interalveolar septum is the wall or portion between two adjacent alveoli. It consists of two thin squamous epithelial layers between which lie capillaries, elastic and reticular fibers and connective tissue, matrix and cells. There are three types of alveolar cells present within the alveoli. These are type 1 pneumocyte, type 2 pneumocyte and alveolar macrophage. Type 1 pneumocyte or squamous alveolar cells, these are 79% of the alveolar surface, extremely thin cell, and the cytoplasm in the thin portion contain the pinocytic vesicles, which may play a role in the turnover of the surfactant and the removal of the small particles containing from the outer surface. In addition to desmosome, all type 1 epithelial cells have occluding junctions that prevent the leakage of tissue fluid into the alveolar air space. The main role of these cells is to provide a barrier of minimal thickness that is readily permeable to gases. Type 2 alveolar cells, that means type 2 pneumocytes, it will cover 3% of the alveolar surface. They are interspread among the type 1 alveolar cells. They are rounded cells that often occur in groups of 2 or 3 along the alveolar surface at point where the alveolar walls unite. These cells rest on the basement membrane and are part of the epithelium. They divided by mitosis to replace their own population and also the type 1 population. In a histologic section, they exhibit a characteristic vesicular or foamy cytoplasm caused by the presence of lamellar bodies which contain phospholipid, glycosaminoglycans and proteins are continuously synthesized and released at the apical surface of the cells. The lamellar bodies give rise to a material that spread over the alveolar surface as the pulmonary surfactant. Function of this cell provides as extracellular coating that lowers the surface tension of the lungs. Alveolar macrophage, this is the third cell, also called the dust cell, are found in the alveoli and in the interalveolar septum. Tens of millions of monocytes migrate daily from the microvasculature into the lung tissue, where they phagocytose erythrocytes lost from damaged capillary and airborne particulates. Now, blood air barrier. Air in the alveoli is separated from capillary blood by the three components referred to collectively as the respiratory membrane or blood air barrier. These are surface lining and cytoplasm of the alveolar cells, fused basal lamina of the closely apposed alveolar cells and capillary endothelial cells and cytoplasm of the endothelial cells. The total thickness of these layers varies from 0.1 to 1.5 micrometer, and function of this barrier permit the diffusion of gases between the alveolar air space and the blood. Oxygen from the alveolar air passes into the capillary blood through the blood air barrier, and carbon dioxide passes from the capillary blood into the alveolus. Capillary endothelial cells are extremely thin and can be easily confused with type 1 alveolar epithelial cell. The endothelial lining of the capillaries is continuous and not fenestrated. Clustering of the nuclei and other organelle allow the remaining areas of the cell to become extremely thin, increasing the efficiency of the gas exchange. The most prominent feature of the cytoplasm in the flattened portion of the cell are numerous pinocytic vesicles. Thank you all. Assalamu alaikum.